Hello YouTube, my name is Neil and we're studying for the MCATs today. We're in the Biochemistry Review, Chapter 10, Carbohydrate Metabolism 2, Aerobic Respiration. Have you ever heard that eating peach pits is deadly? Before you start panicking about the snack you had during your study break, you should know that the accuracy of such a statement is debatable. While it is true that digesting peach pits can result in the formation of trace amounts of cyanide, the concentration is far too low to be clinically worrisome. Cyanide is a poison that binds irreversibly to cytochrome ALA3, a protein located in the electron transport chain of the mitochondria. Why can this be deadly? Blocking the electron transport chain inhibits aerobic respiration from yielding the ATP the body requires to function properly. Cyanide poisoning leaves cells unable to utilize oxygen for aerobic respiration because it blocks oxygen from binding to the ETC. Therefore, symptoms resemble those of tissue hypoxia, perceived difficulty breathing, general weakness, and in higher doses, cardiac arrest followed by death within minutes. But what about the metabolic pathways described in Chapter 9? Don't they produce energy without oxygen? While glycolysis does not depend on oxygen, it only yields a net 2 ATP per molecule of glucose, which is not nearly enough to maintain the body's energy requirements. This brings us to two of the most tested topics on the MCAT, the citric acid cycle and oxidative phosphorylation. In this chapter, we'll take a close look at what's gained when the products of glycolysis and other derivatives of metabolic pathways enter the citric acid cycle. We'll also look at how this process is regulated with regard to the substrates, products, and reactions involved. Lastly, we'll observe what happens when this cycle's products undergo oxidative phosphorylation, with particular emphasis on how the electron transport chain facilitates the process and the ATP that is yielded. Let's just write down that glycolysis yields 2 ATP acetyl-CoA. The citric acid cycle, also called the Krebs cycle or the tricarboxylic acid cycle, occurs in the mitochondria. The main function of this cycle is the oxidation of acetyl-CoA to carbon dioxide and water. In addition, the cycle produces the high-energy electron-carrying molecules NADH and FADH2. Acetyl-CoA can be obtained from the metabolism of carbohydrates, fatty acids, and amino acids, making it a key molecule in the crossroads of many metabolic pathways and a highly testable compound. TCA, tricarboxylic acid, or citric acid, or Krebs cycle. Let's write them all down. Tricarboxylic cycle, Krebs cycle, and citric acid cycle. Acetyl-CoA down to CO2 and water, making NADH and FADH2 in the process. Methods of forming acetyl-CoA. Recall from Chapter 9 that after glucose undergoes glycolysis, its product, pyruvate, enters the mitochondria via active transport and is oxidized and decarboxylated. These reactions are catalyzed by a multi-enzyme complex called the pyruvate Dehydrogenase complex, PDC, which is located in the mitochondrial matrix. As we take a deeper look at the enzymes that make up this complex, as well as the substrates and products of their reactions, it is helpful to follow the carbons in the molecules. For example, the 3-carbon pyruvate is cleaved into 2-carbon acetyl group and carbon dioxide, into a 2-carbon acetyl group and carbon dioxide. This reaction is irreversible, which explains why glucose cannot be formed directly from acetyl-CoA. In mammals, pyruvate dehydrogenase complex is made up of 5 enzymes. Pyruvate dehydrogenase is PDH, dihydrolipol transacetylase, dihydrolipoyl dehydrogenase, pyruvate dehydrogenase kinase, and pyruvate dehydrogenase phosphatase. While the first 3 work in concert to convert pyruvate to acetyl-CoA, the latter 2 regulate the actions of PDH. 10.1 shows the overall reaction for the conversion of pyruvate to acetyl-CoA. The reaction is exergonic. It takes 33.4 kilojoules per mole. The complex is inhibited. It makes 33.4 kilojoules per mole. The complex is inhibited by an accumulation of acetyl-CoA and NADH that can occur if the electron transport chain is not properly functioning or is inhibited. All right. The pyruvate dehydrogenase complex is made up of PDH, pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase, and then pyruvate dehydrogenase kinase, followed by pyruvate dehydrogenase phosphatase. So pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase modify pyruvate dehydrogenase, the first enzyme. And then you have dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase. The first 3, they said, convert pyruvate to acetyl-CoA. The 3-carbon pyruvate is cleaved into 2-carbon acetyl group and carbon dioxide. So pyruvate to acetyl-CoA and carbon dioxide. To acetyl plus CO2. Exergonic. It's irreversible. And it's inhibited by accumulation of acetyl-CoA and NADH. Here we have pyruvate, CoA-SH, NAD+, TPP, lipoate, FAD, magnesium, NADH, and here we have acetyl-CoA in a carbon dioxide molecule. Note that coenzyme A is written as CoA-SH in the reaction above. This is because CoA is a thiol containing an SH group. When acetyl-CoA forms, it does so via covalent attachment of the acetyl group to the SH group, resulting in the formation of a thioester, which contains sulfur instead of the typical oxygen ester OR. The formation of a thioester rather than a typical ester is worth noting because the high energy properties of thioesters, that is to say, when a thioester undergoes a reaction such as hydrolysis, a significant amount of energy will be released. This can be enough to drive other reactions forward, like the citric acid cycle. The pyruvate dehydrogenase complex enzyme needed to catalyze acetyl-CoA formation are listed below in a sequential order, and the mechanism is shown in figure 10.1. So it goes into each of these. PDH, pyruvate dehydrogenase. Pyruvate is oxidized, yielding CO2, while the remaining two carbon molecules bind covalently to thiamine pyrophosphate. TPP. TPP is a coenzyme held by non-covalent interactions to PDH. Magnesium is also required. Seems important. PDH, pyruvate oxidized. Thiamine pyrophosphate is TPP. TPP is a cofactor or coenzyme. Magnesium is the cofactor. Coenzyme. Yielding CO2. Dihydrolipoyl transacetylase. I don't have a nice abbreviation for that. Hydrolipoyl transacetylase. The 2-carbon molecule bonded to TPP is oxidized and transferred to lipoic acid, a coenzyme that is covalently bonded to the enzyme. Lipoic acid's disulfide group acts as an oxidizing agent, creating the acetyl group. The acetyl group is now bounded to lipoic acid via thioester linkage. After this, dihydrolipoyl transacetylase catalyzes the CoA-SH interaction with the newly formed thioester link, causing transfer of an acetyl group to form acetyl-CoA. Lipoic acid is left in its reduced form. So lipoic acid coenzyme thioester link. So let's just write so it transfers lipoic acid, or the acetyl group is bonded to lipoic acid. It transfers it to coenzyme A. So we're just adding an acetyl group to our molecule. To CoA, basically. There's a thioester link. Dihydrolipoyl dehydrogenase. Dihydrolipoyl transacetylase. Not transacetylase, dehydrogenase. Flavin adenine dinucleotide. Flavin adenine dinucleotide. FAD. Is used as a coenzyme in order to reoxidize lipoic acid, allowing lipoic acid to facilitate acetyl-CoA formation in future reactions. As lipoic acid is reoxidized, FAD is reduced to FADH2. In subsequent reactions, this FAD2H2 is reoxidized to FAD, while NAD+ is reduced to NADH. So coenzyme It oxidizes lipoic acid. It is reduced to FADH2, while something else is going to be oxidized. While lipoic acid is oxidized, and when FADH2 is reoxidized, NAD plus is reduced. So FADH2 is reduced, FAD is oxidized, NAD plus is oxidized, NADH is reduced. Just for reference, I should also write that as FAD is reduced, lipoic acid is oxidized to lipoic acid. So you have pyruvate to CO2, pyruvate dehydrogenase, turning acyl TPP to TPP. TPP coenzyme. Acyl TPP. Acyl lipoate. Dihydrolipoyl transacetylase. Coenzyme A to acetyl-CoA. We have lip, which would be our lipoic acid. We have dihydrolipoyl dehydrogenase, which replenishes our FAD. Which replenishes our lipoic acid. Basically, you have pyruvate on this side, coming down to acetyl-CoA on this side. Coenzyme A gets acylated through these two enzymes, I guess. All right, while glycolysis is a heavily reviewed and heavily tested contributor to the production of acetyl-CoA, other pathways are capable of forming acetyl-CoA. These pathways act on fatty acids, ketogenic amino acids, ketone bodies, and alcohol. Descriptions of these pathways are provided below. The ultimate production of acetyl-CoA follows all of these pathways to culminate in the final common pathway of the citric acid cycle. So, fatty acid oxidation or beta oxidation. In the systole, a process called activation causes a thioester bond to form between carboxyl groups of fatty acids and CoA. Because this activated fatty acyl CoA cannot cross the inner mitochondrial membrane, the fatty acyl group is transferred to carnitine via a transesterification reaction. As shown in figure 10.3, carnitine is a molecule that can cross the inner membrane with a fatty acyl group in tow. Once acylcarnitine crosses the inner membrane, it transfers the fatty acyl group to a mitochondrial CoA via another transesterification reaction. In other words, carnitine's function is merely to carry the acyl group from a systolic CoA to a mitochondrial CoA. Once acyl-CoA is formed in the matrix, beta oxidation can occur, which removes 2 carbon fragments from the carboxyl end. So basically, beta oxidation just removes 2 carbons from carboxyl end. And carnitine shuttles it. Transesterification. Amino acid catabolism. Certain amino acids can be used to form acetyl-CoA. These amino acids must lose their amino group via transamination. Their carbon skeletons can then form ketone bodies. These amino acids are termed ketogenic for that reason. The conversion of ketone bodies to acetyl-CoA is mentioned below. Transamination. Transamination to ketone bodies. Ketones is the next group. So amino acids first become ketones. And then ketones, although acetyl-CoA is typically used to produce ketones, when the pyruvate dehydrogenase complex is inhibited, the reverse reaction can occur as well. Ketones can become acetyl-CoA. reversible. Alcohol. When alcohol is consumed in moderate amounts, the enzymes alcohol dehydrogenase and acetaldehyde dehydrogenase convert it to acetyl-CoA. However, this reaction is accompanied by NADH buildup, which inhibits the Krebs cycle. Therefore, the acetyl-CoA formed through this process is primarily used to synthesize fatty acids. They didn't give us a name for the enzyme for amino acids, but for alcohol, they give us alcohol dehydrogenase and acetaldehyde dehydrogenase. NADH buildup inhibits Krebs cycle, or the TCA, so it's used for fatty acids. What is the overall reaction of the pyruvate dehydrogenase complex? The overall reaction is acetyl-CoA to carbon dioxide. No, it's pyruvate to acetyl-CoA. That was this, right? Pyruvate to acetyl-CoA with carbon dioxide, pyruvate and CoA to carbon dioxide and acetyl-CoA. And it also uses an NAD. Like, these are circular, but NAD is not replenished. So if we were to write an overall reaction for this, I think it would look like pyruvate plus coenzyme A plus NAD plus equal or become CO2 plus acetyl-CoA plus NADH. That won't work. Never mind, it's good. And And this fatty acid, figure 10.3, fatty acid activation and transport. It shows the carnitine, the coenzyme A, the activated coenzyme A, and carnitine being able to cross the membrane and turn back into coenzyme A via fatty acyl-CoA synthetase and carnitine acyltransferase, and then a carnitine transporter. 2. What other molecules can be used to make acetyl-CoA, and how does the body perform this conversion for each? Amino acids get turned into ketones. They didn't tell us how ketones This talks about ketones to acetyl-CoA. Amino acids can form acetyl-CoA. These are ketones to acetyl-CoA is the opposite of pyruvate dehydrogenase. Ketone bodies to acetyl-CoA. But they don't tell us what enzyme. The pyruvate dehydrogenase complex is something else, though. Pyruvate to acetyl-CoA. When that's shut down, we'll use this. Well, the mechanism of conversion, so all I could say is ketones become acetyl-CoA. They don't tell us how. In amino acids, they tell us transamination. They don't give us a name. For alcohol, they give us alcohol dehydrogenase and acetaldehyde dehydrogenase. And that's it. Oh, and fatty acids. They remove 2 carbons from the carboxyl end. Once again, they tell us about the carnitine and these enzymes, but they don't tell us oh, this is just to make acetyl-CoA once again. From a fatty acid. The acyl group comes from the fatty acid. CoA to acyl-CoA. But acyl-CoA is not acetyl-CoA, is it? So beta oxidation can occur, which removes 2 carbon fragments from the carboxyl end. Fatty acid plus CoA makes acyl-CoA. But yeah, once again, they don't give us the name of that enzyme. They just give us the name of acyltransferase. So I guess fatty acyl synthetase and then carnitine acyltransferase. Should I write these down? Fatty acyl-CoA and carnitine acyltransferase 1. All right. Let's see what they say. Let's see how they answer these. (10.1) 1. Pyruvate plus CoA plus NAD plus make acetyl-CoA plus CO2 plus NADH. Nice. Oh, plus H. And they take a CoA with the SH on it. That's fine. I think they're the same. 2. Fatty acids shuttle acyl group from systolic CoA to mitochondrial CoA via carnitine, then undergo beta oxidation. Ketogenic amino acids transaminate to lose nitrogen, convert carbon skeleton into ketone body, which can be converted into acetyl-CoA. So it just makes ketones. Ketones reverse the ketone body formation. And once again, they don't really describe it. It's just ketones to acetyl-CoA. Ketone body formation, we did that. Anyways, ketones. Alcohol. Alcohol dehydrogenase and acetaldehyde or acetaldehyde dehydrogenase convert alcohol into acetyl-CoA. Right. Reactions of the citric acid cycle. The citric acid cycle takes place in the mitochondrial matrix and begins with the coupling of a molecule of acetyl-CoA to a molecule of oxaloacetate. While parts of this molecule are oxidized to carbon dioxide and both energy, GTP, and energy carriers, NADH and FADH2, are produced. The other substrates and products of the cycle are reused over and over again. Although oxygen is not directly required in the cycle, the pathway will not occur anaerobically. This is because NADH and FADH2 will accumulate if oxygen is not available for the electron transport chain and will inhibit the cycle. As we look at the individual reactions that take place during the citric acid cycle, it cannot be overemphasized that this process is exactly what it's called, a cycle, not just a series of reactions. An overview of the cycle is provided in figure 10.4, and we'll take a deeper look at those steps below. Start with pyruvate. We get acetyl-CoA, then we get citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate, and then the cycle continues. And we're turning NAD into NADH along the way, and that will be used in the electron transport chain. TCA. Tricarboxylic acid cycle. Acetyl-CoA plus oxaloacetate NAD to NADH like times 3 plus 1 FAD to FADH and 1 GDP to GTP and 2 carbons. So the acetyl-CoA becomes the carbon dioxide. Or it becomes the next oxaloacetate. Let's take a look. Key reactions. 1. Citrate formation. First, acetyl-CoA and oxaloacetate undergo a condensation reaction to form citryl-CoA, an intermediate. Then the hydrolysis of citryl-CoA yields citrate and CoA coenzyme A. This reaction is catalyzed by citrate synthase, as discussed in Chapter 2. Synthases are enzymes that form new covalent bonds without needing significant energy. This second part of this step energetically favors the formation of citrate and helps the cycle revolve in the forward direction. This reaction can be seen in figure 10.5. So we have acetyl-CoA and oxaloacetate make citrate-CoA. So basically the let's see here. We have 1, 2 carbons here. 1, 2, 3, 4 carbons here. Now we have 1, 2, 3, 4, 5, 6 carbons on citrate. So we have a 2 carbon plus a 1 carbon to a 3 carbon. I mean, it's a 6 carbon, but 3 in a chain. Right, we have a 2 carbon and a 4 carbon to a 6 carbon. Let's write acetyl-CoA plus oxaloacetate becomes citrate and CoA, coenzyme A. Step 2, condensation, and then hydrolysis. It's a synthase. Condensation plus hydrolysis. Step 2, citrate isomerized to isocitrate. Isomerization. Achiral citrate is isomerized to one of 4 possible isomers of isocitrate. First citrate binds at 3 points to the enzyme aconitase. Then water is lost from citrate, yielding cis-aconitate. Finally, water is added back to form isocitrate. The enzyme is a metalloprotein that requires iron. In figure 10.6, you can see that this results in a switching of a hydrogen and a hydroxyl group. Overall, this step is necessary to facilitate the subsequent oxidative decarboxylation. A switching of a hydrogen and a hydroxyl. So this hydroxyl goes down here. All right. Step 3. Alpha-ketoglutarate and carbon dioxide formation. Isocitrate is first oxidized to oxalosuccinate by Isocitrate Dehydrogenase. Oh, I wanted to write the enzyme. Aconitase. Aconitase. Three, alpha ketoglutarate and CO2. First we have isocitrate dehydrogenase, which takes isocitrate to oxalosuccinate. Then oxalosuccinate is decarboxylated to produce alpha-ketoglutarate and carbon dioxide, as shown in figure 10.7. This is a very important step to know for test day because isocitrate dehydrogenase is the rate-limiting enzyme of the citric acid cycle. The first two of the carbons from the cycle is lost here. This is also the first NADH produced from intermediates in the cycle. So then oxalosuccinate is decarboxylated to alpha ketoglutarate and CO2. Decarboxylation. Also makes NADH. And it is a rate-limiting step. Here we have isocitrate making oxalosuccinate, and then alpha-ketoglutarate with carbon dioxide. So we got 1, 2, 3, 4, 5 carbons instead of 6. We lost our first carbon. We started with 4 carbons in oxaloacetate. In the end, we're going to end up with 4 carbons for oxaloacetate for the beginning of the next cycle. It's really 2 carbons here that we've broken one of them off. To get energy out of. Step 4 is succinyl-CoA and carbon dioxide. Succinyl-CoA and CO2. These reactions are carried out by the alpha-ketoglutarate dehydrogenase complex, which is similar in mechanism, cofactors, and coenzymes to the pyruvate dehydrogenase complex. In the formation of succinyl-CoA, alpha-ketoglutarate and coenzyme A come together and produce a molecule of carbon dioxide as shown in figure 10.8. This carbon dioxide represents the second and last carbon lost from the cycle. Reducing NAD products produces another NADH. So alpha-ketoglutarate to succinyl-CoA plus CO2 via alpha-ketoglutarate dehydrogenase complex with TPP, lipoic acid, and magnesium as cofactors, just like PDH. And we also get that NADH from NAD. And we also use a coenzyme A. So alpha-ketoglutarate plus CoA. All right, step 5. Succinate formation. Hydrolysis of the thioester on succinyl-CoA yields succinate and CoA and is coupled to the phosphorylation of GDP to GTP. This reaction is catalyzed by succinyl-CoA synthetase, as shown in figure 10.9. Synthetases, unlike synthases, create new covalent bonds with energy input. Recall the earlier discussion about thioester bonds with regard to acetyl-CoA. They're unique in their hydrolysis is accompanied by a significant release of energy. Therefore, phosphorylation of GDP to GTP is driven by the energy released by thioester hydrolysis. Once GTP is formed, an enzyme called nucleoside diphosphate kinase catalyzes phosphate transfer from GTP to ADP, thus producing ATP. Note that this is the only time in the citric acid cycle that ATP is produced directly. ATP production occurs predominantly within the electron transport chain. Succinate formation. So, succinyl CoA to succinate and coenzyme A is coupled to phosphorylation of GTP. So it releases energy. Reaction is catalyzed by succinyl CoA synthase. And then GTP becomes ATP. I guess it probably just becomes GDP again, but that is coupled to ADP to ATP by nucleoside diphosphate kinase. All right. We have a little diagram of succinyl CoA turning into succinate and GDP turning into GTP and making coenzyme A. So our coenzyme A from this step we get back. Then step 6, fumarate. This is the only step of the citric acid cycle that doesn't take place in the mitochondrial matrix. Instead, it occurs on the inner membrane. Let's look at why. Succinate undergoes oxidation to yield fumarate. This reaction is catalyzed by succinate dehydrogenase. Succinate dehydrogenase is considered a flavoprotein because it is covalently bonded to FAD, the electron acceptor in this reaction. This enzyme is an integral protein on the inner mitochondrial membrane. As succinate is oxidized to fumarate, FAD is reduced to FADH2. Each molecule of FADH2 then passes the electrons it carries to the electron transport chain, which eventually leads to the production of 1.5 ATP, unlike NADH, which will give rise to 2.5 ATP. FAD is the electron acceptor in this reaction because the reducing power of succinate is not great enough to reduce NAD+. So we have succinate to fumarate by succinate dehydrogenase. takes FAD, H2. It's in the mitochondrial matrix, or it's in the it's in the inner membrane. Mitochondrial inner membrane. It's a flavoprotein. Malate Formation, 7. The enzyme fumarate catalyzes the hydrolysis of the alkene bond in fumarate, thereby giving rise to malate. Although 2 enantiomeric forms are possible, only L-malate forms in this reaction. Fumarase takes fumarate to malate. Oxaloacetate formed anew. Step 8. Oxaloacetate. The enzyme Malate Dehydrogenase catalyzes the oxidation of malate to oxaloacetate. A third and final molecule of NAD+ is reduced to NADH. The newly formed oxaloacetate is ready to take part in another turn of the citric acid cycle. And we've gained all of the high-energy electron carriers possible from one turn of the cycle. The last steps of the citric acid cycle from succinate to oxaloacetate are shown in figure 10.10. So we have succinate dehydrogenase, fumarase, malate dehydrogenase, the last step. Malate to oxaloacetate via malate dehydrogenase. Use up another NAD. Net results in ATP yield. Now let's take a step back and see what our net yield is from the steps we just took. Starting with the pyruvate dehydrogenase complex, recall that the products of this reaction include 1 acetyl-CoA and 1 NADH. In the citric acid cycle, steps 3, 4, and 8 each produce 1 NADH, while step 6 forms 1 FADH2. Step 5 yields 1 GTP, which can be converted to ATP. 2 carbons leave the cycle in the form of CO2. Each NADH can be converted to approximately 2.5 ATP, while each FADH2 molecule can yield about 1.5 ATP. The total amount of chemical energy harvested per pyruvate is listed below. ATP production. 4 NADH makes 10 ATP. 1 FADH2 makes 1.5 ATP. 1 GTP is an ATP. The total is 12.5 ATP per pyruvate, which is 25 ATP per glucose. Glycolysis yields 2 ATP and 2 NADH, providing another 7 molecules of ATP. Thus, the net yield of ATP for one glucose molecule from glycolysis through oxidative phosphorylation is 30 to 32 ATP. Note that the efficiency of glycolysis varies slightly from cell to cell, so there's a range of ATP yield from one molecule of glucose. So one glucose is 2 pyruvate and there's 12.5 ATP per pyruvate, 25 ATP per glucose. So this is just the oxidative phosphorylation part. This is just the TCA. Glycolysis is 2 ATP and 2 NADH, and each NADH is 2.5 ATP, so that's 5 ATP plus 2 is 7, so glycolysis makes 7 ATP. This isn't glucose, this is pyruvate. Oh no, it is 25 for glucose. $25 plus 7 is 32. I thought glycolysis takes glucose down to pyruvate. And this is pyruvate. Glucose to pyruvate, and pyruvate down to acetyl-CoA, and then carbon dioxides eventually, and NAD. So 12.5 plus 12.5. 25 plus 7. Total of 32 ATP. Regulation. Let's say it's test day and you see the following question. Which of the following is an inhibitor of isocitrate dehydrogenase? Before you start to panic, take a step back and use critical thinking. Where have we heard of Isocitrate Dehydrogenase or merely isocitrate before? The Krebs cycle. By knowing this, you can already make a fair attempt at such a question. Because energy, ATP, and energy carriers, NADH and FADH2, are products of this process, it makes sense that these molecules would give a negative feedback effect on the citric acid cycle. Always consider the big picture when faced with questions like these. For now, we'll outline how regulation occurs throughout the citric acid cycle. Look for the recurring theme that energy products inhibit energy production processes. Energy products inhibit energy production processes. All right, pyruvate dehydrogenase complex regulation. Regulation of PDH complex. Even upstream from its actual starting point, the citric acid cycle can be regulated. The mechanism by which this happens is phosphorylation of PDH, which is facilitated by the enzyme pyruvate dehydrogenase kinase. Thus, whenever levels of ATP rise, phosphorylating PDH inhibits acetyl-CoA production. Conversely, the pyruvate dehydrogenase complex is reactivated by the enzyme pyruvate dehydrogenase phosphatase in response to high levels of ADP. By removing a phosphate from PDH, pyruvate dehydrogenase phosphatase is able to reactivate acetyl-CoA production. Acetyl-CoA also has a negative feedback effect on its own production. When using alternative fuel sources such as fats, the acetyl-CoA production is sufficient to make it redundant to continue producing acetyl-CoA from carbohydrate metabolism. That's part of why eating a high-fat meal fills you up so quickly. ATP and NADH, as markers of the cell being satisfied energetically, also inhibit PDH. So ATP and acetyl-CoA. ATP, NADH, and acetyl-CoA all inhibit. Control points of the citric acid cycle. There are 3 essential checkpoints that regulate the citric acid cycle from within, and allosteric activators and inhibitors regulate all of them. The details of these mechanisms are outlined below and in Figure 1011. Citrate synthetase, or synthase, ATP and NADH function as allosteric inhibitors of citrate synthase, which makes sense because both are products, indirect and direct, respectively, of the enzyme. Citrate also allosterically inhibits citrate synthase directly. So does succinyl-CoA. Citrate as well as succinyl-CoA. Should we write these down? Should. We wrote PDH. Now, in the TCA cycle, citrate synthase inhibited by ATP, NADH, citrate, and succinyl CoA, which is downstream Isocentrate Dehydrogenase. Isocitrate Dehydrogenase is inhibited by ATP and NADH. Alpha ketoglutarate dehydrogenase complex, succinyl CoA, NADH, ATP is inhibitory. Note that high levels of ATP and NADH inhibit the citric acid cycle, while high levels of ADP and NAD+ promote it. This isn't a coincidence. When energy is being consumed in large amounts, more and more ATP is converted to ADP and NADH is converted to NAD+. It is therefore the ATP to ADP ratio and NADH to NAD+ ratio that helps determine whether the citric acid cycle will be inhibited or activated. During a metabolically active state, ADP and NAD+ levels should rise as ATP and NADH levels decline, thus inducing activation at all the various checkpoints described above, replacing the energy used up by active tissues. What is the purpose of all the reactions that collectively make up the citric acid cycle? It's to use the carbon bonds in acetyl-CoA to make NADH for the most part. What enzyme catalyzes the rate-limiting step of the citric acid cycle? The rate-limiting step. Is that the first step? The PDH, Pyruvate Dehydrogenase complex? Where did I write rate limiting? I wrote it down somewhere. Rate limiting. Alpha ketoglutarate is the rate limiting step, right? Alpha ketoglutarate production. So isocitrate dehydrogenase, isocitrate to alpha ketoglutarate, specifically the decarboxylation of oxalosuccinate, is the rate limiting step. So it's inhibited by ATP, NADH. It's stimulated by ADP and NAD+. What are the three main sites of regulation within the citric acid cycle? What molecules inhibit and activate the three main checkpoints? So the inhibitors and activators are always ATP and NADH for the inhibitors and ADP and NAD+ for the activators. The checkpoints would be Isocitrate Dehydrogenase, alpha-ketoglutarate dehydrogenase, and then Succinate Dehydrogenase, as well as Malate Dehydrogenase, all the dehydrogenases, I guess. Malate dehydrogenase, succinate dehydrogenase, alpha-ketoglutarate dehydrogenase, and isocitrate dehydrogenase. Interesting, all dehydrogenases. He said 3. I see like 5, including up here. 1, 2, 3, 4. So these are steps requiring NAD and FAD. But inhibition is at acetyl-CoA and oxaloacetate. That's the PDH, right? That's the pyruvate. No, pyruvate dehydrogenase makes acetyl-CoA. Acetyl-CoA plus oxaloacetate. Is a synthase. Citrate synthase. Oh, it's inhibited by citrate. The alpha-ketoglutarate dehydrogenase is inhibited by succinyl-CoA. Citrate synthase is also inhibited by succinyl-CoA. The isocitrate dehydrogenase is just inhibited by ATP and NADH. And stimulated by ADP and NAD+. There's no stimulated for the other two. Let's see what they say about it. One, complete oxidation of carbons in intermediates to CO2 so that reduction reactions can be coupled with CO2 formation, thus forming energy carriers such as NADH and FADH2 for the electron transport chain. Right. 2, isocitrate dehydrogenase is the key rate-limiting step. Isocitrate dehydrogenase. Right. Key enzymes or key steps of regulation: citrate synthase, ATP and NADH, Succinyl-CoA and citrate are all inhibitors, while activators, there are none. Isocitrate dehydrogenase is inhibited by ATP and NADH and activated by ADP and NAD+. Alpha-ketoglutarate dehydrogenase, they just call it alpha-glutarate complex, is inhibited by ATP, NADH, and succinyl-CoA, while it's activated by ADP and calcium. That is not what it says here, but there you go, calcium as well. So let's put promoters in green and inhibitors in red. And let's add some of these to these key steps. Isocitrate dehydrogenase seems like the key. Oh no, alpha-ketoglutarate dehydrogenase. No, isocitrate was the one. Isocitrate dehydrogenase is upregulated by ADP and NAD+, downregulated by ATP and NADH, citrate synthase is upregulated by ATP. Oh, it's inhibited rather. By ATP, NADH, Succinyl-CoA, and citrate. Alpha-ketoglutarate dehydrogenase is also inhibited by ATP, NADH, Succinyl-CoA, and upregulated by ADP and calcium. All right. .3 is the electron transport chain. All right. The ETC The electron transport chain is the final common pathway that utilizes the harvested electrons from different fuels in the body. It is important to make the distinction that it is not the flow of electrons but the proton gradient it generates that ultimately produces ATP. Aerobic metabolism is the most efficient way of generating energy in living systems, and the mitochondrion is the reason why. In eukaryotes, the aerobic components of respiration are executed in mitochondria, while anaerobic processes such as glycolysis and fermentation occur in the systole. Looking at figure 1012, notice how the components of the mitochondria are critical in the harvesting of energy. The citric acid cycle takes place in the mitochondrial matrix. The assemblies needed to complete oxidative phosphorylation are housed adjacent to the matrix in the inner membrane of the mitochondria. The inner mitochondrial membrane is assembled into folds called cristae, which maximize surface area. It is the inner mitochondrial membrane that will be essential for generating ATP using the proton motive force, an electrochemical proton gradient generated by the complexes of the electron transport chain. The final step in aerobic respiration is actually two steps: electron transport along the inner mitochondrial membrane and the generation of ATP via ADP phosphorylation. While these two processes are actually separate entities, they are very much coupled, so explaining these steps together makes a great deal of sense. The electron-rich molecules NADH and FADH2 are formed as byproducts at earlier steps in respiration. They transfer their electrons to carrier proteins located along the inner mitochondrial membrane. Finally, these electrons are given to oxygen in the form of hydride ions, and water is formed. While this is happening, energy released from transporting electrons facilitates proton transport at three specific locations in the chain. Protons are moved from the mitochondrial matrix into the inner membrane space of the mitochondria, thereby creating a greater concentration gradient of hydrogen ions that can be used to drive ATP production. Electron flow in complexes. Should we write down that proteins in inner membrane and the high concentration of protons is in the matrix, the mitochondrial matrix. Protons are moved from the mitochondrial matrix into the inner membrane space, thereby creating a greater concentration gradient of hydrogen ions that can be used to drive. So the hydrogens are in inner membrane space. Electron flow in complexes. The formation of ATP is endergonic. The electron transport is an exergonic pathway. By coupling these reactions, the energy yielded by one reaction can fuel the other. In order for energy to be harnessed via electron transport reactions, the proteins along the inner membrane must transfer the electrons donated by NADH and FADH2 in a specific order and direction. The physical property that determines the direction of electron flow is reduction potential. Recall from chapter 12 in chemistry that if you pair two molecules with different reduction potentials, the molecule with the higher potential will be reduced while the other molecule becomes oxidized. The electron transport chain is therefore nothing more than a series of oxidations and reductions that occur via the same mechanism. NADH is a good electron donor, and the high reduction potential of oxygen makes it a great final acceptor in the electron transport chain. The organizational structure of the membrane-bound complexes that make up the transport chain is diagrammed in figure 1013 and further detailed below. Complex 1, complex 2, complex 3, complex 4, and we have NAD to NADH to NAD, and that's worth 4 protons plus 2 protons here at the end. Succinate to fumarate is worth 2 protons plus another 2 protons. Complex 3 has 2 steps. Step 1 is worth 2 protons. Step 2 is worth 2 protons. And then finally, complex 4. Looks like it actually takes protons. It moves 2 across. But it looks like it uses up 4 to move 2 across. Let's see. We'll get into it in a second. Complex 1. It says step 1 and 2 of complex 3 are drawn as 2 separate steps. Here for clarity, however, the same COQH2, cytochrome C, oxidoreductase complex is used for both steps. But first, complex 1 is NADH, CoQ oxidoreductase. NADH, CoQ oxidoreductase. The transfer of electrons from NADH to coenzyme Q is catalyzed in this first complex. This complex has over 20 subunits, but the 2 highlighted here include a protein that has an iron-sulfur cluster and a flavoprotein that oxidizes NADH. The flavoprotein has a coenzyme called flavin mononucleotide, FMN, covalently bonded to it. FMN is quite similar in structure to FAD, flavin adenine dinucleotide. The first step in the reaction involves NADH transferring its electrons over to FMN, thereby becoming oxidized to NAD+, as FMN is reduced to FMNH2. Next, the flavoprotein becomes reoxidized while the iron-sulfur subunit is reduced. Finally, the reduced iron-sulfur subunit donates the electrons it receives from FMNH2 to coenzyme Q, also called ubiquinone. Coenzyme Q becomes CoQH2. This first complex is one of 3 sites where proton pumping occurs as 4 protons are moved to the inner membrane space. So first, NAD is oxidized. So I'm going to write oxidized on top, NADH. As the electrons, it's going to get oxidized to NAD+. And FMN is reduced. So FMN is reduced to FMNH2. I'm going to write oxidized and reduced down here. FMN is reduced Next, the flavoprotein becomes reoxidized while the iron-sulfur subunit is reduced. I'm just going to write the iron subunit as Fe. So this is reoxidized as Fe is reduced. I guess it's FeS. Finally, the reduced iron subunit donates the electrons it receives from FMNH to coenzyme Q. So, coenzyme Q becomes CoQH2. Negative and a positive. I'm going to write it in blue because I don't know if it's technical, but it's the oxidized and reduced form. I don't know if it has a hydrogen on it or just like a lone charge. Let's see, they just write FeS reduced and FeS oxidized. This will be the same thing, positive and negative charges. And I put the charges on the wrong one. This is positive, this is negative. All right. The net effect is passing high energy electrons from NADH to CoQ to form CoQH2. All right, that's all complex 1. Complex 2, succinate CoQ oxidoreductase. Succinate CoQ oxidoreductase. Just like complex 1, complex 2 transfers electrons to coenzyme Q, while complex 1 receives electrons from NADH. Complex 2 actually receives electrons from succinate. Remember that succinate is a citric acid cycle intermediate and that it is oxidized to fumarate upon interacting with FAD. FAD is covalently bonded to complex 2, and once succinate is oxidized, it's converted to FADH2. After this, FADH2 gets reoxidized to FAD as it reduces an iron-sulfur protein. The final step reoxidizes the iron-sulfur protein as coenzyme Q is reduced. Because succinate dehydrogenase was responsible for oxidizing succinate to fumarate in the citric acid cycle, it makes sense that succinate dehydrogenase is also part of complex 2. It should be noted that no hydrogen pumping occurs here to contribute to the proton gradient. So yeah, the whole point of this is 2 protons move across. And the net effect of all of this is 2 protons. The net effect of this is no protons. Succinate CoQ oxidoreductase from succinate. So here we'll start with succinate being oxidized to fumarate. Reducing FAD, FADH2, and then FADH2 gets reoxidized to FAD when our iron sulfur gets reduced. The final step, iron is reoxidized as CoQ is once again reduced to CoQH2. No protons. No protons. Here we had 2 protons go across. H2, H plus. Complex 3. No protons. Complex 3 is CoQH2 Cytochrome C oxidoreductase, also called cytochrome reductase. This complex facilitates the transfer of electrons from coenzyme Q to cytochrome C in a few steps through complex 3. Though complex 3 is drawn as 2 separate complexes in figure 1013 to illustrate the sequential reactions which occur within the complex, both of these steps are occurring within the same complex, using the same coenzyme Q. The overall reaction is written below. The following steps involve the oxidation and reduction of cytochromes, proteins with heme groups in which iron is reduced to Fe2+ and reoxidized to Fe3+. So between 3 and 2. So this is probably also between 3 and 2. In the transfer of electrons from iron, only one electron is transferred per reaction. But because coenzyme Q has 2 electrons to transfer, 2 cytochrome C molecules will be needed. Complex 3's main contribution to the proton motive force is via the Q cycle. In the Q cycle, 2 electrons are shuttled from a molecule of ubiquinol, C CoQ-H2, near the intermembrane space to a molecule of ubiquinone, CoQ, near the mitochondrial matrix. Another 2 electrons are attached to heme moieties, reducing 2 molecules of cytochrome C. A carrier containing iron and sulfur assists this process. In shuttling these electrons, 4 protons are also displaced to the intermembrane space. Therefore, the Q cycle continues to increase the gradient of the proton motive force across the inner mitochondrial membrane. So cytochrome C is reduced So a total of 4 protons, 2 for each of the steps. And it looks like each step reduces the cytochrome C. So let's write yeah, so I guess in this case, CoQ-H2 is oxidized to CoQ, and cytochrome C is reduced. We have this times 2. X. So our protons are 2 times 2. All right. Complex 4 is cytochrome C oxidase. This complex facilitates the culminating step of the electron transport chain, transfer of electrons from cytochrome C to oxygen, the final electron acceptor. This complex includes subunits of cytochrome A, cytochrome A3, and copper 2+ ions. Together, cytochromes A and A3 make up cytochrome oxidase. Through a series of redox reactions, cytochrome oxidase gets oxidized as oxygen becomes reduced and forms water. This is the final location on the transport chain where proton pumping occurs, as two protons are moved across the membrane. The role proton pumping plays in ATP synthesis is an essential one that we will describe in detail next. The overall reaction is 4 cytochrome C plus 4 hydrogens plus oxygen makes 4 cytochrome C in its oxidized form plus 2 H2O. H2O is reduced. Oxygen is reduced. So cytochrome C let's write down cytochrome A and A3 get oxidized while oxygen gets reduced. And 2 protons move across. All right, proton motive force. Let's take a step back and look at the proton gradient that formed as electrons were passed along the ETC. As a concentration of protons increases in the inner membrane space, two things happen simultaneously. pH drops in the inner membrane space, and the voltage difference between the inner membrane space and the matrix increases due to proton pumping. Together, these two changes contribute to what is referred to as an electrochemical gradient, a gradient that has both chemical and electrostatic properties. Because it is based on protons, we often refer to the electrochemical gradient across the inner mitochondrial membrane as the proton motive force. An electrochemical gradient stores energy, and it will be the responsibility of ATP synthase to harness this energy to form ATP and ADP and an inorganic phosphate. ATP synthase. NADH shuttles. As we look at the net ATP yield per glucose, note that a range exists between 30 and 32. This is because efficiency of aerobic respiration varies between cells. This variable efficiency is caused by the fact that systolic NADH formed through glycolysis cannot directly cross into the mitochondrial matrix. Because it cannot contribute its electrons to the transport chain directly, it must find alternate means of transportation referred to as shuttle mechanisms. A shuttle mechanism transfers the high-energy electrons of NADH to a carrier that can cross the inner mitochondrial membrane. Depending on which of the two shuttle mechanisms NADH participates in, either 1.5 or 2.5 ATP will end up being produced. Let's take a look at the two mechanisms. guess let's just write down that ATP synthase takes ADP and PPI, or is it just PI? Not pyrophosphate, but inorganic phosphate, to make ATP. NADH shuttle, like the glycerol 3 phosphate shuttle. The systol contains one isoform of glycerol 3 phosphate dehydrogenase, which oxidizes systolic NADH to NAD+, while forming glycerol 3 phosphate from dihydroxyacetone phosphate, DHAP. On the outer surface of the inner mitochondrial membrane, there exists another isoform of glycerol 3 phosphate dehydrogenase that is FAD dependent. This mitochondrial FAD is the oxidizing agent and ends up being reduced to FADH2. Once reduced, FADH2 proceeds to transfer its electrons to the ETC via complex 2, thus generating 1.5 ATP for each molecule of systolic NADH that participates in this pathway, which is shown in figure 1014. So we have NADH being oxidized while a DHAP is reduced to G3P, and then G3P is going to turn back into DHAP, while FAD is reduced to FADH2, and then finally FADH2 is returned to FAD as Q is quinol, CoQ. Is that what it is? Is Q quinol? It's not CoQ, it's just quinone. CoQ is quinone, so maybe CoQ and Q are the same? Once reduced, FADH2 proceeds to transfer its electrons to the ETC via complex 2. So this is complex 2 at the bottom. Some of these other things here are GPDH, GPDHC, and GPDHM. right. The malate aspartate shuttle shuttles. They're all NADH shuttles. This one is malate aspartate. Systolic oxaloacetate, which cannot pass through the inner mitochondrial membrane, is reduced to malate, which can. This is accomplished by systolic malate dehydrogenase. Accompanying this reduction is the oxidation of systolic NADH to NAD+. Once malate crosses into the matrix, mitochondrial malate dehydrogenase reverses the reaction to form mitochondrial NADH. Now that NADH is in the matrix, it can pass along its electrons to the ETC via complex 1 and generates 2.5 ATP per molecule of NADH. Recycling the malate requires oxidation to oxaloacetate, which can be transaminated to form aspartate. Aspartate crosses into the systole and can be converted to oxaloacetate to restart the cycle. So here, this is oxidized and this is reduced. This is oxidized. Oxaloacetate is reduced. Sorry, so this is being reduced. This is being oxidized. Here, NADH is being reduced. So malate is being oxidized, oxidized, reduced, reduced. Malate aspartate. Aspartate is reduced to oxaloacetate. Is further reduced to malate by aspartate transaminase. I'll write this over again. aspartate transaminase and oxaloacetate to malate by malate dehydrogenase, which incidentally also turns malate back into oxaloacetate, and aspartate transaminase also turns oxaloacetate back into aspartate in the mitochondria. So these can now go across the membrane. Oxaloacetate can't. And of course, here NADH becomes NAD plus. Here NAD plus becomes NADH. And here alpha ketoglutarate becomes glutamate, and here the opposite. Glutamate becomes alpha ketoglutarate. All right. And this is some kind of membrane. Mitochondrial membrane. And don't forget that this transfers its NAD, will transfer its electrons, NADH. Will transfer its electrons to complex 1. All right. Which complexes are associated with each of the following? Circle all that apply. Proton, pumping a proton into the intermembrane space. 1, not 2, but 3 and 4 all do it. Acquiring electrons from NADH. 1 and just 1 does it? Just 1. Acquiring electrons from FADH2. Complex 2 does that. Having the highest reduction potential. I guess the 2 times 2 would be complex 3. Having the highest reduction potential. I guess, yeah. Number of protons also is going to be the same as reduction potential. All right, 2. What role does the electron transport chain play in the generation of ATP? It provides the proton motive force to pump protons across the inner mitochondrial membrane, making a electrochemical gradient that ATP synthase uses to make ATP. 3, based on its needs, which of the 2 shuttle mechanisms is cardiac muscle most likely to utilize and why? That's right. That's an important point. While the first one is capable of producing 1.5 ATP, the malate aspartate shuttle is capable of making 2.5 ATP. And why? Because the difference in reducing power of FAD and NAD, I guess, is what it comes down to. FAD is just a little less capable, I guess. All right. 10.4, oxidative phosphorylation. We have arrived at the payout site of aerobic respiration, ATP synthesis. Knowing the nuances of ATP synthesis is an absolute must by test day. The link between electron transport and ATP synthesis starts with a protein complex called ATP synthase, which spans the entire inner mitochondrial membrane and protrudes into the matrix. Chemiosmotic coupling. The proton motive force interacts with the portion of ATP synthase that spans the membrane, which is called the F0 portion, F sub 0. F0 functions as an ion channel, so protons travel through F0 along their gradient back into the matrix. As this happens, a process called chemiosmotic coupling allows the chemical energy of the gradient to be harnessed as a means of phosphorylating ADP, thus forming ATP. In other words, the ETC generates a high concentration of protons in the inner membrane space, which then flows through F0 ion channel of ATP synthase back into the matrix. As this happens, the other portion of ATP synthase, which is called the F0 portion, utilizes the energy released from this electrochemical gradient to phosphorylate ADP to ATP. So demonstrated in figure 1016. The specific mechanism by which ADP is actually phosphorylated is still a matter of debate. Chemiosmotic coupling. Is oxidative force phosphorylation the entire thing or just oxidative phosphorylation? Is the whole system reducing oxygen to phosphorylate ADP? So we have the F0 ion channel. And it takes ADP to ATP. Chemiosmotic coupling describes a direct relationship between the proton gradient and ATP synthesis. It is a predominant mechanism accepted in the scientific community when describing oxidative phosphorylation. However, another mechanism called conformational coupling suggests that the relationship between the proton gradient and ATP synthesis is indirect. Instead, ATP is released by the synthase as a result of conformational change caused by the gradient. In this mechanism, the F1 portion of ATP synthase is reminiscent of a turbine spinning within a stationary compartment to facilitate the harnessing of gradient energy for chemical bonding. So conformational chemiosmotic coupling is the F0 portion. Conformational coupling would be the theory that the F1 portion is causing conformational changes. F1 is like a turbine. ATP synthase generates ATP from ADP and inorganic phosphate by allowing high-energy protons to move down the concentration gradient created by the electron transport chain. So we know now how we generate ATP, but how much energy was required to do so? When the proton motive force is dissipated through the F0 portion of ATP synthase, the free energy change of the reaction is negative 220 kilojoules per mole, a highly exergonic reaction. This makes sense because phosphorylating ADP to form ATP is an endergonic process, so by coupling these reactions, the energy harnessed from one reaction can drive another. Regulation. Because the citric acid cycle provides the electron-rich molecules that feed into the ETC, it should come as no surprise that the rates of oxidative phosphorylation in the citric acid cycle are closely coordinated. Always think of O2 and ADP as the key regulators of oxidative phosphorylation. If O2 is limited, the rate of oxidative phosphorylation decreases and the concentrations of NADH and FADH2 increase. The accumulation of NADH in turn inhibits the citric acid cycle. The coordinated regulation of these pathways is known as respiratory control. In the presence of adequate O2, the rate of oxidative phosphorylation is dependent on the availability of ADP. The concentration of ADP and ATP are reciprocally related. An accumulation of ADP is accompanied by a decrease in ATP and the amount of energy available to the cell. Therefore, ADP accumulation signals the need for ATP synthesis. ADP allosterically activates isocitrate dehydrogenase, thereby increasing the rate of the citric acid cycle and the production of NADH and FADH2. The elevated levels of these reduced coenzymes in turn increase the rate of electron transport and ATP synthesis. Respiratory control. O2 promotes O2 ADP promote so let's say if O2 is low, then NADH and FADH2 will be high, which inhibits the TCA. All right. Similarly, increase in ADP. All right, what is the difference between ETC and oxidative phosphorylation? What links the two? The ETC is the part that sets up the proton motive force, the electrochemical gradient, and then the oxidative phosphorylation is kind of the part that uses that gradient to make ATP. 2, the delta G of NADH reducing oxygen directly is significantly greater than any individual step along the electron transport chain. If this is the case, why does transferring electrons along the ETC generate more ATP than direct reduction of oxygen by NADH? Because each step in the ETC is using different molecules, different NADHs, succinates, or cytochromes, to build up more proton motive force than just the reduction of one oxygen. I guess. Let's see what they say. One, the ETC is made up of the physical set of inner membrane proteins located on the inner mitochondrial matrix, and they undergo oxidation reduction reactions as they transfer electrons to oxygen. The final electron acceptor. As electrons are transferred, a proton motive force is generated in the inner membrane space. Oxidative phosphorylation is the process by which ATP is generated via harnessing the proton gradient, and it utilizes ATP synthase to do so. 2, by splitting up electron transfer into several complexes, enough energy is released to facilitate the creation of a proton gradient at many locations rather than just one. The greater the proton gradient, the greater the ATP generation will be. Direct reduction of oxygen by NADH would release a significant amount of energy to the environment, resulting in inefficient electron transport. So releasing energy into the environment is not what we want to do. We don't just want to make heat, per se. Although each of these reactions should still make some heat, right? All right, then we have discrete practice questions. Number 1, during a myocardial infarction, the oxygen supply to an area of the heart is dramatically reduced, forcing the cardiac myocytes to switch to anaerobic metabolism. Under these conditions, which of the following enzymes would be activated by increased levels of intracellular AMP? So, anaerobic metabolism Succinate Dehydrogenase is not anaerobic, right? Phosphofructokinase is. Isocitrate Dehydrogenase isn't. Pyruvate Dehydrogenase. All the dehydrogenases we learned about in oxidative phosphorylation, aerobic respiration, and anaerobic respiration, that's from the last chapter. The only one of these that's from the last chapter is PFKD. 2. A patient has been exposed to a toxic compound that increases the permeability of mitochondrial membranes to protons. Which of the following metabolic changes would be expected in this patient? Increases permeability, the proton motive force, the electrochemical gradient is going to be less effective. So they'll have increased ATP synthase activity, no. Increased ATP levels, no. Decreased pyruvate dehydrogenase activity, or increased oxygen utilization. You could still have increased oxygen utilization. Because the oxygen part is in the ETC. Or decreased pyruvate dehydrogenase activity. If the proton motive force is ineffective, you won't get the metabolites. So it's not like you won't get the ATP buildup. So ATP won't be able to inhibit earlier enzymes in the pathway like pyruvate dehydrogenase. So I think increased oxygen utilization sounds like the right answer. Which of the following incorrectly pairs metabolic process with its site of occurrence? Glycolysis in the systole, citric acid cycle in the outer mitochondrial membrane, ATP phosphorylation in the systole and mitochondria, electron transport chain, inner mitochondrial membrane. Electron transport chain is along the inner mitochondrial membrane. ATP phosphorylation happens in the mitochondria. Does it happen in the systole? Glycolysis does produce a couple ATP. So I think that fits, that's fine. Citric acid cycle in the outer mitochondrial membrane. Does that happen in the outer mitochondrial membrane? It's inside, right? Occurs in the mitochondria. PDC is located in the mitochondrial matrix. So the matrix is the inner layer of the mitochondria, not the inner membrane space. So not the outer mitochondrial membrane. I think B is the one that's incorrect. Let's verify that by asking Google the location of citric acid cycle. The matrix of the mitochondria. Yeah, the mitochondrial matrix is the inner layer. The inner membrane, intermembrane space would be outer mitochondrial membrane. Right. So, number 4. Which of the following processes has the following net reaction? 2-acetyl-CoA plus 6-NAD plus 2-FAD plus 2-GDP plus 2 pyrophosphates, not pyrophosphates, inorganic phosphates, and 6 H2Os make 4 CO2, 6 NADH, 2 FADH2, 2 GTP, 6 hydrogens, and 2 CoAs, coenzyme A's. Acetyl-CoA to CoA. Acetyl-CoA so I guess it's the tricarboxylic acid as a whole. It's not pyruvate. We start with acetyl-CoA, tricarboxylic acid cycle. That is the TCA cycle. Four is going to be C. Is it the TCA? Yeah. TCA cycle. All right. 4 is C. 5, in glucose degradation under aerobic conditions, oxygen is the final electron acceptor. Oxygen is necessary for all ATP synthesis. Net water is consumed. The proton motive force is necessary for all ATP synthesis. So when they say for all ATP synthesis, there's a couple ATP made by glycolysis. So even under aerobic conditions, the first steps of taking glucose down to pyruvate is going to make 2 ATP. So B and D can't be right. And between A and C, is water consumed? I'm not sure. I do know that oxygen is the final electron acceptor. The reduction of oxygen to water. That kind of implies the creation of water, not the consumption of water. But it makes sense that water is used in other places too. So not dehydrogenase, hydrolysis uses water. So are there any hydrolysis steps? They're mostly dehydrogenase steps. Anyways, 5, it's got to be that oxygen is the final electron acceptor. 6, fatty acids enter the catabolic pathway in the form of glycerol, adipose tissue, acetyl-CoA, or ketone bodies. Fatty acids make let's see, where is this? Other places for acetyl-CoA to come from. Beta oxidation. makes acyl-CoA. Fatty acids enter the catabolic pathway in the form of ketone bodies, adipose tissue, glycerol. Is it glycerol? We go back to beta oxidation. These pathways act on fatty acids, ketogenic amino acids, ketone bodies, and alcohol. Describing these pathways below, the beta oxidation, fatty acyl-CoA, or CoA. So if it's not acyl-CoA, I'll go with acetyl-CoA. Yeah, I'd say it's acetyl-CoA. I'll go with C. In which of the following reactions is the reactant oxidized? FAD to FADH2, that's reduced. NAD plus to NADH, it's also reduced. NADPH to NAD plus, that might be oxidized. ADP to ATP, I guess that's reduced. NAD plus is useful, NADH is less useful, right? NAD plus is reduced and it oxidizes something else. So NADPH to NAD plus is oxidation. But between ADP and ATP, which one is more oxidized? It says that ADP and ATP aren't considered oxidized of each other. Phosphorylation of ADP doesn't equal oxidation. All right, so 7's got to be C. 8. In which part of the cell is cytochrome C located? The mitochondrial matrix, the outer mitochondrial membrane, the inner mitochondrial membrane, or the systole? Well, from the looks of this diagram, while this is the inner membrane, this is the inner membrane, this is the inner space, that's the intermembrane space. This is the matrix, this is the inner membrane space. And it looks like these cytochromes are in the inner membrane space. So I wouldn't say that they're in the mitochondrial matrix. Yeah, they're pumping the protons out of the matrix. So it looks like they're not in the matrix, they're not in the systole. Inner mitochondrial membrane or outer mitochondrial membrane? It's the inner mitochondrial membrane. It's part of that system. So let's go C9. Which of the following correctly shows the amount of ATP produced from the given high energy carriers? FADH2 make 1.5, NADH make 2.5. So NADH 1.5. And they have, oh no, this one is NADH. FADH2 is worth 1.5 ATP. 9 is B. 10. Why is it preferable to cleave thioester links rather than typical ester links in aerobic metabolism? Oxygen must be conserved for the electron transport chain. Thioester hydrolysis has a higher energy yield, which sounds good. B. C. Typical ester hydrolysis can occur in vivo. Or D. Thioester cleavage requires more energy. Thioester hydrolysis has a higher energy yield, so we could use it to make an ATP. 11. Which enzyme converts GDP to GTP? So it does use hydrolysis, so it does use water, right? I was wondering about whether these processes use water or not. 11. Which enzyme converts GDP to GTP? Nucleoside diphosphate phosphatase, nucleoside diphosphate kinase, isocitrate dehydrogenase, pyruvate dehydrogenase. GDP to GTP would be a phosphatase. So I want to say nucleoside phosphatase. But I want to look at my part of the TCA cycle where GDP turns into GTP because of succinyl-CoA synthetase. That is not one of the options here. A step where ATP is made, first GDP is made. And that's not one of the options, so I think the phosphatase has got to be it. Kinase cleaves phosphatase phosphorylates, right? Phosphatase that removes a phosphate group. My mistake. Kinase adds a phosphate group. A kinase transfers from ATP to a specific molecule, but ATP can itself be kinase, right? But phosphatase definitely isn't it. Which is the phosphatase and which is the kinase? Yeah. Phosphatase just cleaves a pyrophosphate, or a inorganic phosphate. So that's not making ATP. It's got to be the kinase that actually makes the ATP. Well, I'm going to go with B instead for 11. We'll see. 12. Which of the following best explains why systolic NADH can yield potentially less ATP than mitochondrial NADH? Systolic NADH always loses energy when transferring electrons. Once NADH enters the matrix from the systole, it becomes FADH2. Electron transfer from systole to matrix can take more than one pathway. There's an energy cost for bringing systolic NADH into the matrix. Yeah, that makes sense. D. 13. In high doses, aspirin functions as a mitochondrial uncoupler. How would this affect glycogen stores? Mitochondrial decoupler. So would it deplete glycogen stores? It has no effect on glycogen stores. It promotes additional storage of glucose, or it affects on glycogen stores varies from cell to cell. Well, if you're not getting the oxidative phosphorylation from mitochondria, and you're just depending on glycolysis and anaerobic respiration, I bet you would deplete your glycogen. It could vary from cell to cell. Like, most cells might deplete glycogen, but your liver might start packaging it up for other cells to get. So maybe D is more accurate. Which complex does not contribute to the proton motive force? I think it was 2 that wasn't contributing. B. 15. Which of the following directly provides the energy needed to form ATP in the mitochondria? The electron transfer in the electron transport chain, an electrochemical gradient, proton gradient, oxidation of acetyl-CoA, or beta oxidation of fatty acids. The most direct is the electrochemical proton gradient. B. Let's see what the answers are. 1 is B, 2 is B, 3 is B, 4 is C, 5 is A, 6 is C, 7 is C, 8 is C, 9 is B, 10 is B, 11 is B, 12 is C after all, not D. The words of these answers choices is critical. The electrons from systolic NADH can enter the mitochondria through one of 2 shuttle mechanisms. The glycerol 3-phosphate shuttle, which ultimately moves these electrons to mitochondrial FAD, and the malate aspartate shuttle, which ultimately moves these electrons to mitochondrial NAD+. If the electrons are transferred using the malate aspartate shuttle, then no energy is lost, making choice A and D incorrect. NADH cannot enter the matrix directly, making choice B incorrect. It is the fact that electrons can use more than one pathway, one of which loses energy that could be used for ATP synthesis, that accounts for the potentially decreased yield of ATP from systolic NADH. So because it could take more than one pathway, that's why it could potentially yield less ATP. In some pathways, there is no energy cost. So depending on whether it takes that pathway or not, you'll have a varying cost. So 12 is C, 13 is A, not D. Uncouplers inhibit ATP synthesis without affecting the electron transport chain. Because the body must burn more fuel to maintain the proton motive force, glycogen stores will be mobilized to feed into glycolysis. Then the TCA, and finally oxidative phosphorylation. So different effects on different cells. That's not really the point. Most cells, we're ignoring like the liver, I guess. Even in the liver, you have to burn glucose. It causes depletion of glycogen stores. If you're not using oxidative phosphorylation, you're going to have to get glucose. You're going to have to use glycogen because glucose, you're still going to, the products of glycolysis aren't going to be further used. So glycolysis is the only thing that's producing ATP. So you're just going to need more glycolysis. So you're going to need more glucose. So you're going to take it from glycogen. 14 is going to be B, and 15 is also B. There you have it. That is Carbohydrate Metabolism 2, Aerobic Respiration. Next time, Chapter 11, Lipid and Amino Acid Metabolism. Thanks for watching. I'll see you next time.